Genes act as the genetic material. Now it has been established. How it was established? That the gene has a role for its expression, and how that expression is being brought about at the phenotypic level. It first came into the knowledge of Garrod in 1902. He was studying a lot many human diseases, and he had established that it is the link between the metabolism and gene which is the key to gene expression. He had said that genes have their expression in the form that they have direct effect on the metabolism of the cell. That is how genes express themselves. So, we are going to find out how genes are expressing themselves in metabolism. Now, with time in 1950s around, there was an experiment that was carried out by Beadle and Totem on the fungi Neurospora crassa. This fungi is known as Drosophila of plant kingdom, just a name, a title it has got. They studied this particular fungi, and in this study they had studied the ornithine citrulline arginine cycle. Now, this is a process known as ornithine cycle. In this, what they did is they took this Neurospora crassa. It is a metabolic process in which ornithine is getting converted to citrulline. Citrulline is further converted to arginine. Now, what they did is they irradiated with this Neurospora crassa with some X-rays and got mutants. There were certain mutants which survived on arginine, all of them. They could survive in arginine. That means arginine was the final product of the metabolic pathway. Right. Because the mutation takes place at the genetic level, we have studied that when the mutation arises, it is directly affecting the genes, right. So, there must be some alteration in the gene. Because the mutants, certain of them survived, all of them survived in arginine, but few of them survived in ornithine containing medium, few of them survived in citrulline containing medium. They carried this particular this experiment in 3 stages. There were certain mutants which survived in arginine condition. Type 1 mutant, they survived in arginine, but at the same time they survived in citrulline as well, they survived in ornithine as well. Alright. Or you can put it other way, they didn't survive in ornithine, they didn't survive in citrulline. The other ones didn't survive in ornithine, but they survived in citrulline and arginine. The last ones, they survived in all the conditions, all the mediums which contained ornithine, which contained citrulline, which contained arginine. Now, what we get an idea from over here is that because this is a metabolic process, it would be guided by certain enzyme at these stages. There are 3 types of mutants which were obtained as a result of irradiation by X-rays of Neurospora crassa. The first class of mutants, they survived in arginine. The second class, they survived in citrulline and arginine both. The third class, they survived in everything. Now, at every stage you are needing the enzymes. That means those which did not survive in ornithine when provided with ornithine, when provided with citrulline, but when given arginine, they survived. Okay, so arginine being the final product, but if only given ornithine, they could not metabolize into citrulline, then citrulline could not be metabolized into arginine, and because they did not have the final product, because of that, the mutants did not survive only in the material containing ornithine. They did not survive in the medium containing only citrulline, but they do survive in arginine because arginine is the final product. They could not metabolize this, that is why they did not survive. Now, this is the class of mutants which are surviving in ornithine as well. They could metabolize ornithine to citrulline, citrulline to arginine, they are getting the final product. So, this experiment in a way showed that when the mutation takes place, it is affecting this stage. The mutation occurring at gene level has affected this stage. Now, what is this stage? This stage is nothing but enzymatic activity. That means the gene has direct effect on metabolism by affecting the enzyme. These mutants, they did not have the enzyme that was required at this stage. That means the metabolism guiding principle of genes is that they affect enzymes. Now, how do they affect? Supposedly, mutation has come. When we studied mutation, we said that it totally changes the code which is responsible for production of something. That means the enzyme is not being formed. So, it led to the proposition of one gene, one enzyme hypothesis. Later on, with the detailed studies of biomolecules, it was concluded that all enzymes are proteins, but not all proteins are enzymes. So, it was replaced and it said one gene, one protein. Later on, it was concluded that it is not only protein, it is one gene, one polypeptide, which could either form an enzyme, which could either form a protein, secondary, tertiary, quaternary, whatever it could be. So, this experiment carried by these two people proposed that one gene controls the production of a polypeptide, which could be playing an important role in the metabolic process stages. This is how the gene expresses itself. That means we have something to do with the formation of proteins. Let's use the word proteins instead of polypeptides. Now, this DNA, which is a gene, this gene contains DNA. The nucleotide has to form proteins. How those proteins are being formed, that is part of next two lessons.